Hi, good afternoon. Am I audible?
Yes, sir.
Okay. So, hi, I'm Rishabh, and we'll be starting with the week 3 of discussion and with the RNA biology classes. So, yesterday we started with the nucleotide metabolism, that is the biochemistry part in the RNA biology, and we completed the de novo pathway of nucleotide synthesis. In this, we studied that how both pyrimidine and purines were synthesized on a ribose uh, backbone, and the basic difference between the salvage and the de novo pathway is that in de novo pathway the nucleotides are synthesized from scratch, and in the salvage pathway the nucleotides are basically recycled. When after degradation some of the bases are formed and then they are recycled and joined to the ribose unit forming the nucleotides. So we discussed the de novo pathway in detail yesterday, and today we'll be discussing about the salvage pathway. So the basic need of why the salvage pathway is that we use PRPP, that is a precursor for the ribose 5 phosphate. So it basically yields ribose 5 phosphate, which attaches with the base for forming the nucleotide. So this is particularly the simplest pathway for synthesis of nucleotides. So, just a brief introduction. So, as we discussed, that the ribose is attached to the preformed nitrogenous base that are formed by the degradation of the nucleotides. These nucleotides or different nucleic acids can be either the nucleic acids of the own body, and the nucleic acids that come from our food. Okay, so this is the source for the nitrogenous bases. Now, after that, when ribose, the bases are attached to the ribose, the phosphate is attached to them, forming the nucleotide, the final molecule. And like I said, the nucleic acids or their derivatives in diet can be used in the formation of the bases. And where this salvage pathway is important, so it is basically involved in every cell, and it's a supplementary pathway of nucleotide synthesis. So uh, why this is a supplementary pathway? Because the major pathway of nucleotide formation is the de novo pathway. So this is the second supplementary pathway which utilizes all the bases that are already in degradation or that the body has already left out, so that we can reuse those bases. Now, this pathway is important in erythrocytes because in erythrocytes, the de novo pathway cannot operate because of the absence of PRPP amidotransferase that transfers the amide group from the glutamine amino acid to the base. So, basically, where there, due to the absence of this PRPP amidotransferase, the de novo pathway cannot operate. That leads to the shift of whole the nucleotide synthesis through salvage pathway. Although erythrocytes do not need that much amount of nucleotide synthesis, but for energy metabolism and all those things, the erythrocytes need the nucleotide synthesis. So salvage pathway is the major pathway that is performed in the erythrocytes. Okay. So I forgot to ask, have you studied the de novo pathway prior?
Yes, sir.
Okay, then it would not be difficult for you to understand this. So first we'll study with the pyrimidine biosynthesis. So what happens here, where so as we know, pyrimidines are basically cytosine, thymine, and uracil. So basically for uracil, first we'll study the uracil is directly attached to the PRPP, that is phosphoribosyl pyrophosphate. So here, what happens? The uracil is directly transferred to the ribose 5-phosphate wala part, and the using the enzyme for uracil phosphoribosyl transferase. So now, which forms the UMP, and pyrophosphate is released. Okay, so this is the simplest uh, conversion that happens. Now, similarly for orotate, that is the pyrimidine part or the precursor for the pyrimidines. So, for any addition of the orotate to the PRPP, the orotate phosphoribosyl transferase act into picture, and then it transfers the orotate molecule to the PRPP molecule, forming pyrimidine monophosphate, releasing the pyrophosphates. So this is the simplest pathway for the formation of the pyrimidine nucleotides. Okay, so moving on to the purine biosynthesis. So here in purine biosynthesis, what happens? We have adenine. So purines are adenine and guanine. Okay, so in adenine, what happens? The same way the PRPP is involved for the source of ribose 5-phosphate. So here adenine interacts with the PRPP and releases PPI, forming AMP in the presence of the enzyme adenine phosphoribosyl transferase. So in Salvage pathway, the basic difference is the use of the type of enzyme that is involved. Like in uracil, that was uracil phosphoribosyl transferase, or in cytosine we have cytosine phosphoribosyl transferase. So similarly in adenine we have adenine phosphoribosyl transferase. So this leads to the formation of AMP. And secondly, for guanine, actually, when guanine is formed, it is formed from the oxo or the keto precursor of guanine, that is xanthine and hypoxanthine. The structure of these two we'll study in the next slide, where degradation of nucleic acid we'll study. And so what happens? This guanine is converted to GMP by the addition of ribose 5-phosphate using the enzyme HGPRT, or hypoxanthine guanine phosphoribosyl transferase. So in short, we'll use HGPRT. Okay. So what happens? This enzyme also catalyzes the conversion of hypoxanthine to IMP, that is inosine monophosphate. So this is the same enzyme HGPRT involved in the conversion of hypoxanthine to the formation of nucleotide IMP. Okay, so this is the basic salvage pathway for the purine biosynthesis. Okay, so do you have any doubt till now? Okay, then moving on. So, like, we'll study a bit more. So, the in metabolism, the deficiency of this particular enzyme, okay, leads to a syndrome called as Leishman syndrome. Okay, so what happens during the deficiency or when there is a mutation in this particular gene, the HGPRT enzyme goes down. So this leads to the accumulation of the hypoxanthine and all those preformed bases in the brain. That leads to poorly coordinated and intellectual defects in children that are less than 2 years old. Okay, so this is the basic characteristics of the Leishman syndrome, that is that when HGPRT is absent, that leads to accumulation of guanine, hypoxanthine, or xanthine in the brain, leading to poor coordination. And this syndrome is basically recognized in the children that are less than 2 years of age. Shall I move on? Okay, now we'll be moving on to the nucleotide degradation part, that is the nucleotide catabolism. So, first we'll study with the purine catabolism wala part. So we have two nucleotides here, GMP, guanosine monophosphate, and AMP, that is adenosine monophosphate. So first, what happens in any of these reactions, the phosphate is removed from the nucleotides. Okay, so that is basically done by the 5' nucleotidase, because as you know that the phosphate group is attached to the 5' carbon end. So first this bond is broken and phosphate is released. So that is done with the help of the enzyme 5' nucleotidase. So in GMP, first the inorganic phosphate is released and then this guanosine is formed. This is a nucleoside. So in this nucleoside, then using nucleosidase, the ribose is removed. That leaves with only the guanine base. Okay. Now after this guanine is formed, then the amino group, that is this particular group, is removed using guanine deaminase, leading to the formation of xanthine, that is the keto form, because of this particular keto group. Okay, so this is the sixth carbon of guanine. Now after this, xanthine oxidase comes into play, and that leads to via oxidation reaction with the use of oxygen, it releases hydrogen peroxide, releasing the uric acid. So this is the major catabolic product of the purine catabolism. This is the question asked in various competitive examinations, that what is the major product of the catabolism, that of nucleotides. So this uric acid is the major product of the catabolism of nucleotides. So this is the degradation of the guanine monophosphate. So starting with GMP, first the phosphate is released, then ribose is released, leading to guanine. Then deamination of guanine takes place, leading to the formation of xanthine, that is the keto form, and After xanthine, via xanthine oxidase, uric acid is formed. Okay. Now, in AMP, the similar process happens, but the intermediates are different. So, in AMP, first the phosphate is released using the same enzyme, 5' nucleotidase. Then, after this, adenosine is formed, that is the nucleoside of the adenine. And after this, the uh, amino group of the adenine is removed by the enzyme adenosine deaminase. So, here it is changed to inosine. Okay. Now, after this, the ribose is removed, leading to the formation of hypoxanthine. That is the keto form for adenine. Okay. Now, this hypoxanthine is converted to xanthine. That is a common intermediate for both the GMP as well as AMP degradation. So, this is catalyzed by the enzyme xanthine oxidase only, and then the xanthine is formed, and xanthine is converted to uric acid. So this is the basic pathway of degradation for the adenine monophosphate. So here the things that are asked in the competitive examination is that inosine is a derivative of adenine, as well as adenine is a derivative of inosine. So both are interrelated to each other. And then xanthine oxidase is the major oxidase used in both the cycles of catabolism. Further, if we see that this enzyme adenosine deaminase is an important enzyme in the body, and any mutations in the gene encoding this particular enzyme leads to a disease called as SCID, that is severe combined immunodeficiency. And what happens, the there is no such cure in this, and the T and B cells of the body immune system are basically affected during the deficiency of this particular disease, because during lifetime these two cells are the only cells that are basically formed, and different genome arrangement happens during the developmental stage, and these cells cannot be formed during the later period of the immune after the immune system is compromised. So the current therapy for SCID is gene therapy only for now, where the functional gene is being introduced into the patients, leading to the uh, repair of the immune system. Okay, and the second important enzyme is the xanthine oxidase. So, in if xanthine oxidase is basically mutated in any of the diseases, that leads to the accumulation of uric acid. So, uric acid accumulation leads to gout. So, gout can be two types. First is the using uh, the related to the urea cycle, where urea is generated very much in the body, and second is related to the nucleotide metabolism. So, this is basically leading to the formation of uric acid crystals in the joints. So, that leads to joint pain. And the current treatment is that allopurinol, that is basically a anti-metabolite. So it leads to the stoppage of inhibition of the xanthine oxidase, so that uric acid is not formed in the body, leading to some relief to the patient suffering from gout.
Sir, hyperuricemia and gout are same thing or they are different?
No, hyperuricemia is basically when the urea levels are high in the body. So that can be due to the nitrogen metabolism in the body. But yes, if some other issue can arise, and hyperuricemia can lead to gout also. So that some cases are also being reported where the hyperuricemia leads to gout, but they are not the same thing.
Okay, sir.
So is this pathway clear?
Yes, sir.
Okay, now moving on to the pyrimidine catabolism. So, in pyrimidine catabolism, what we see, we have two kinds of pathway. One is the for thymine, and the other one is for the cytosine. So, first we'll see with the cytosine one. So, what happens? Like similar to the guanine and adenine, first the amino component of the cytosine is removed using a cytosine deaminase, forming uracil. Then after that, uracil is converted to dihydrouracil with the help of the this is a redox reaction involving NADPH, because two hydrogen will be used in this particular reaction. Then the N-carbamoyl beta-alanine is formed in this process, forming beta-alanine. That is basically used in uh, protein catabolism, protein metabolism. So, and here one molecule of carbon dioxide and ammonia are also released. Then, talking about thymine, so what happens in thymine? First, the dihydrosyl dehydrogenase enzyme comes and it basically degrades it to dihydrothymine. So, similar to uracil, first uracil is converted to dihydrouracil, the thymine is converted to dihydrothymine by the reduction of this particular bond. Then using dihydropyrimidase, this compound is the chain is broken down, and this beta uh, uridoso butyrate is formed. Now further, this particular enzymes uh, degrades this compound and form amino isobutyrate, releasing the ammonia and the bicarbonate ions. So further, this amino transferase, like with the help of the alpha ketoglutarate, it basically forms the methyl malonyl semialdehyde that leads to the formation of succinyl coenzyme. So this succinyl coenzyme helps in fatty acid synthesis, and sometimes it is also involved in valine synthesis during this intermediate pathway of valine synthesis. So the thymine catabolism is basically a four-step process with complex intermediates, and similar to the cytosine degradation. So here the byproducts for cytosine, direct question is asked after the cytosine degradation or uracil degradation, how many byproducts are formed. So for cytosine, there will be two molecules of ammonia, carbon dioxide, and alanine. And if you are talking about the uracil, so in uracil, only one molecule of ammonia will be formed. So these are the products of the cytosine and uracil degradation. And for thymine, here for thymine, one molecule of ammonia, then CO2, and in the last, it forms either valine or succinic acid. So basically, this methyl melanosemidaldehyde is the product of the thymine catabolism. Okay, so is this pathway clear?
Yes, sir.
Okay, so here we complete the nucleotide metabolism wala part, and now we'll see some of the questions that were asked in various competitive examinations in the past years. So, starting with this particular question. So, aspartate is an amino acid with the structure. This is the structure of the aspartic acid, and given below are the biosynthetic processes occurring in cells. So, is it involved in protein synthesis, de novo synthesis of inosine monophosphate and orotic acid, synthesis of adenosine monophosphate from inosine monophosphate, glutathione synthesis. So, which of the following options correctly represents all the biosynthetic process wherein aspartate is involved as a precursor? So, can you think about the answer?
Sir, in salvage pathway of pyrimidine synthesis.
Hmm. Sorry, I didn't get the question.
Sir, aspartate
Hmm.
is a precursor of which will um, form, sir, CTP, I think.
Uh, Sorry, which option did you say?
Sir, can you repeat the options? Sir,
Okay.
I'm unable to see
Okay, so you are not able to see the question.
the.
Is the question visible now?
Yes, yes. So option four, sir. I'm only sure about the B part,
Hmm.
that is de novo synthesis of ionosin, and that is only present in option fourth. That's why I chose fourth, but I'm not sure about it is protein synthesis. How it is related to glutathione synthesis, sir? Can you please explain?
Okay. So yes, it's not involved in glutathione synthesis. Okay, so that's why the option is like given for confusion only. And like aspartate is an amino acid, right? So it will be definitely involved in protein synthesis. So for sure, first is the correct option. Now going to the de novo synthesis of inosine monophosphate and orotic acid. Okay, so what happens during a de novo synthesis of pyrimidine metabolite, if you remember the structure. So I'm basically drawing the l- simplest structure. So what happens, this particular so in case of pyrimidine, this particular end is provided by the aspartate. Okay, and in pyrimidine, in purines, what happens? Hmm. So this is so this particular N, that is nitrogen, is also provided by the aspartic acid. So in case of uh, purines, the intermediate that is formed is inosine monophosphate. And in case of pyrimidines, the intermediate that is formed prior to the formation of the nucleotide is orotate or orotic acid. Okay, so here the this particular nitrogens of both the purine and pyrimidine is provided by aspartate. Okay, so that's why the option B is correct. And going to option 3, so synthesis of adenosine monophosphate from inosine monophosphate. So yes, what happens in when inosine monophosphate is formed? So then it converts to adenosine monophosphate and guanosine monophosphate. So this adenosine monophosphates require a additional amino group at the sixth position. So that is provided basically by the aspartate residue. So option C is also correct. So the correct option is option D, that is A, B, and C. So is the explanation clear, or shall I show you with the diagrams?
Sir, can you please represent it with a diagram?
Okay, so then I'll open that. Just give me 2 minutes. Do you able to see the screen?
Yes, sir.
Okay. So this is the de novo pathway for pyrimidine synthesis. Just 1 minute. So what happens during pyrimidine synthesis de novo pathway? The aspartate residue basically acts with the carbamoyl phosphate and forms this kind of structure. Okay, so this is the first nitrogen that is provided by the aspartate residue. Okay, or shall I explain the whole pathway?
No, no, sir.
Hmm.
Got it.
Okay, so this is the pyrimidine synthesis. Now comes the purine wala part. So can you able to see the slide for purine synthesis?
Yes, sir. Yes, sir.
Okay. So in this eighth step, okay. So here first what happens in purines, the imidazole ring, that is the five-membered ring, is formed first, and then addition to that the aspartate is added, and this forms the first carbon. Okay, so this is the first carbon of the purine ring, and here the aspartate adds its amine group. Okay, so here the in formation of the IMP, so aspartate is involved in the addition of the first nitrogen. Okay, and this was the de novo wala option. So de novo aspartate is involved in the de novo pathway for the synthesis of inosine monophosphate as well as orotic acid. So
Okay, okay,
now
sir.
moving on to the third option that we have for the conversion of inosine monophosphate to the adenosine monophosphate. So here, as you can see, so this inosine monophosphate adds the aspartate residue and adds a amino group at the uh, like along with the purine side chain. Okay, so this amino group basically represents the adenine. So this aspartate also helps in the formation of adenine monophosphate. Okay,
Yes,
so
sir. Yes, sir.
this is with the third option. So is it now clear that how these two options are there?
Yes, sir. Yes, sir.
Okay. So now moving on to the next question. Now, is the question visible to you?
Not yet, sir.
Okay. Now is it
Yes,
visible?
yes, sir. Yes.
Hmm. So, how many carbons of the purine ring are contributed by the folate, one carbon pool during purine biosynthesis? So, the options are 0, 1, 2, 3. So, can you guess?
No, sir. Please
Mhm.
elaborate. I don't get.
Okay. So, in purines, um do you remember the basic structure of the purine ring?
Yes, sir.
Okay. So, what happens? I'll draw once and then we'll see. So, okay, I'm not involving the double bonds for now for simplicity, but this is the basic structure of the purine ring.
Sir, it
Starting
is not
from,
visible.
sorry,
Sir, it is not visible.
oh, is it visible now?
Yes, sir.
Okay. So this is the basic, the ring part is visible now that I have drawn.
Ah, yes, sir.
Okay, so this is the basic ring of the purine that we see, and I have not involved the double bond and all those structures for simplicity. Okay, so what we have, we first name these particular atoms. So one, This is the first carbon, second, third, fourth, fifth, sixth, seventh, eighth, ninth. Okay, so just to remember, in pyrimidines, we have two nitrogen atoms at first and third positions, and in purines, we have 1, 3, 7, and 9. Okay, so this is for simplicity to remember, so that you can remember the entire ring structure when needed. Now, in this particular purine residues, the first nitrogen, as we saw, is provided by the aspartate residue. Okay, the these three are provided by the glycine amino acid. The fourth, fifth carbon and the seventh nitrogen is provided by the glycine amino acid. The third and ninth nitrogen is provided by the glutamine molecule. Okay. The sixth carbon is provided by the carbon dioxide molecule that is formed during our catabolism process.
Yes, sir.
And the second and the eighth carbon is basically provided by the folate molecule.
Got it, sir.
Okay, so two molecules of carbon are in basically provided by the folate molecule during purine biosynthesis. So the correct answer is option 2. Oh, sorry, option 3. Okay. I have uploaded the yesterday's presentation in the Drive link, and I'll soon be sharing the recording of the lecture also. You can see that. And if you have any
Yes, sir.
doubt, you can ask in the next lecture. Okay. So, is there any doubt in this particular question?
No, sir. No, sir.
Okay. Now, similar, I'll explain what the for the pyrimidine also. So, pyrimidine have a ring of six atoms. So, starting with this. Okay, so this is first, second, third, fourth, fifth, and sixth. So here the first nitrogen from here till here is provided by the uh, wait a minute. Hmm, sorry. So from here to here, it is provided by the aspartate molecule. Okay, this nitrogen actually these two compounds are basically provided by the carbamyl phosphate, and carbamyl phosphate is basically formed from glutamine and bicarbonate ion. So this N is provided by the glutamine molecule, and this carbon is provided by bicarbonate ion. Okay, so this is basic uh, sharing of atoms from different molecules in the pyrimidine chain. Okay.
Yes, sir.
Hmm. So let's move on to the next question. So what I just gave you explanation in the last slide. So this is the same question. So in the biosynthesis of purine, this is a purine diagram that is given. So you have to select the correct option. So all N atoms, C4 and C5 from aspartic acid, N1 is from aspartic acid, N3 and N9 are from glutamine side chain, N7, C4 and C5 from glycine, N1 is from aspartic acid, N3 from glutamine side chain, N9 from N attached C alpha of glutamine, or N7, C4 and C5 from glycine, And the last option, N1 is from glutamine, N3 is from glutamine side chain, N9 is from the N attached to C alpha of glutamine, and N7 C4 C5 from glycine. So can you now answer?
Sir, I'm confused between option number 2 and 3.
Okay. So what's the confusion?
Uh, Sir, N9 of from
Hmm.
N attached to calcium of glutamine is
Calcium.
the confusion. Yes, sir.
Hmm. Okay. So N attached to the C alpha, that is the major or the anomeric carbon of the amino acid. So this is basically the amine group that they are talking about. So nitrogen involved in the amine group of the glutamine. So is it now clear what this means?
Yes, sir.
Hmm. So is the confusion sorted out now, or do you need more explanation?
Which one is correct between both of them?
Okay. So what happens when glutamine is involved in any of these? It basically transfer its amide group. Okay, that is the side chain of the amino acid. Okay, so here in option 3, first we'll see. So like N1 from aspartic acid, that is fine. N3 is from the glutamine side chain, that is the amide group. N9 from the N attached to C alpha of glutamine, that is the amino group of the glutamine. Okay, and N7 C4 C5 and from glycine. Okay, so these three from glycine. So this one is correct. This one is correct. This one is correct. Only the N9 part, that is not provided by the glutamine side. That is provided by the glutamine side chain, not the amino group of the glutamine. Okay, so this leads to that option 2 is correct.
Got it, sir.
Okay, and there is one more like difference here for aspartate. Okay, because it has only one amino group, not because the side chain is carboxylic acid. So here the N provided by the aspartate is the amino group. So can you see my screen?
Yes, sir.
Hmm. So the next question is, which of the following contributes nitrogen atoms to both purine and pyrimidine rings? Okay, so the options are carbamoyl phosphate, aspartate, carbon dioxide, or glutamine.
Sir, aspartate.
Hmm. Okay, so aspartate is correct. Then we just discussed about glutamine also.
Yes, sir, glutamine.
So both of the amino acids are involved in providing the nitrogen atoms in both purine and pyrimidine rings. Okay, so the answer is
Yes,
aspartate
sir.
and glutamine.
Okay, sir.
Hmm. Okay, so let's move on to the next question. Uh, d- are you able to see my screen?
No, sir, it is the
Stuck.
previous question.
Okay. Hmm. Can you now able to see the screen?
Yes, sir.
Hmm. So the carbon in CO2 is labeled with the heavy isotope C13. So which of the atoms of purine and pyrimidine ring will be labeled with C13? C6 of the pyrimidine ring, C2 of the pyrimidine ring, C4 of the purine ring, or C8 of the purine ring? Just think and answer.
Sir, option B.
C2 of the pyrimidine ring. Okay.
Uh, Answer in fluorine, carbon number 6 will be
Hmm.
C13.
Yeah. So yes, the correct answer is option B. And in purine ring, we have discussed this now itself, that the carbon dioxide provides, or the bicarbonate ions. Okay, this provides basically one carbon in the pyrimidine ring, that is the C2, this one, and sorry, this one, and in the purine ring, so this one, this is the sixth carbon. So sixth carbon in the purine ring and the second carbon in the pyrimidine ring is provided by the carbon dioxide. So if we label C it as C13, so this carbon will be labeled as C13 and this carbon will be labeled as C13. Okay. So this leads to the end of the nucleotide metabolism wala part. And now we'll move on to the next part of the biochemistry. So till here, is there any doubt and or anything that you need to ask about any of these processes? You can ask.
No, sir, I'll study first, then
Hmm.
I'll ask.
Okay, sure. Now, we'll move on to the next part, that is RNA being the catalytic molecule, the basic structure of the ribozyme. Today, I'll just touch upon the basics and how it leads to the formation of the ribozyme. And on in the next class, we'll discuss some of the examples of the catalytic ribosome, catalytic RNA units. So, starting with the basic RNA world hypothesis basically states that, so We have studied the RNA world hypothesis. So this hypothesis basically states that the RNA molecule can have two roles. That is, it can act as a self-replicating molecule and also can have some catalytic role. Okay, so this catalytic role is basically like there is a molecule being named after RNA, that is ribozyme. That is not a protein component, but the RNA only performing the catalytic function. So the catalytic role for RNA is being taken on from the discovery of the catalytic RNA, the ribozymes, in 1980s. So this is the first evidence of being RNA being the catalytic role. Now the second evidence come from the ribosomes. Okay, so ribosomes that are formed in the nucleolus have two components. One is the protein part and other is the RNA part. Rather than RNA, we'll say ribosomal RNA part. And ribosome is formed by the combination of both proteins as well as rRNAs, forming a tertiary or a quaternary structure of a mechanism. And in this particular ribosome also helps in protein synthesis. Okay, so that helps in basically translating the mRNA to a protein. So this is the second evidence for RNA having a catalytic role. Although it has a combination conjugation with the protein molecule, but it also has a catalytic role. Okay, now comes the third evidence that artificially, like some scientists like constructed some artificial RNA constructs, and they found that key in those constructs can catalyze any of the reaction in the body. Okay, so this artificial RNA constructs behaved basically like proteins. And then the last evidence came from the sequences of RNA that are capable of simple form of self replication. This example is being taken for from the transposomes. Okay, so these are basically the evidences that suggest that RNA can have a catalytic role also instead of proteins. So we consider proteins only as the enzymes or performing the catalytic role, but here it also provides the evidence that RNA can also have some of the catalytic roles. Okay, so any doubt till now?
No, sir.
Okay, so now this leads to the possible remnants of the RNA world. For example, the RNase P ribozyme that catalyzes the tRNA precursor. Okay, so this basically is the enzyme that during tRNA formation there is splicing and all those processing going on. So there is a molecule called RNase P that catalyzes the uh, degradation of the precursor molecule and formation of the mature tRNA molecule that functions as a adapter molecule, the self splicing introns. So we have during splicing we'll study that in some of the introns, for example we have spliceosomes or the group 1 introns. So group 1 introns has the capability of splicing on their own. Okay, so they form a lariat kind of structure where the intron is basically removed out of the exon. So this is the exon, and based on the sequences present in the intron, this particular intron is removed and a complex L-shaped structure is formed while they are removal. Okay, so this particular reaction requires no protein, hence they can take out on their own from this particular region. While in case of spliceosomes, the RNAs are but basically combined with the proteins, like in case of the ribosomes, forming a spliceosome complex. Okay, now then there are self-cleaving viral RNAs. So there are some viruses that form these RNAs that cleave the nucleic acids formed as a defense mechanism. Thus, they pass from the defense mechanism of the body using these particular self-cleaving viral RNAs. Then the famous example of peptide transferase in the ribosome. So as we discussed that ribosome has like the peptide transferase activity is performed by 23S rRNA and the 28 and 5.8S rRNA in eukaryotes. So this one is for the prokaryote one and this one is for the eukaryote one. Okay, and then comes the last point, the nucleotides involved in the metabolism and signaling, etc. So this is the basically example from the triphosphates that we have, like ATP, GTP, and all those molecules that are involved in the metabolism and the signaling, that is CMP. Okay, in addition to triphosphates, we have coenzyme A, the redox reaction NADP, NADH, and then we have FADH. So these molecules also have an adenine moiety that are considered to be a precursor. Sorry, RNA is being a precursor of these molecules, and then CMP that is involved in the secondary messenger in signaling. So these examples show us that like RNA world is not only an hypothesis, but it also has some of the current examples in our body. Okay, is it clear?
Yes, sir.
Okay, so now moving on. So the ribozyme, it is basically an RNA that has a catalytic role, and it is not a protein. So it was this is it this discovery got a Nobel Prize in 1989 in chemistry to Sidney Altman and Thomas Say for discovery of RNA in living cells that is not only a molecule of heredity but also function as a biocatalyst. So this was an important discovery of that time and yielded a Nobel Prize. So as we know that proteins have structure. For example, it has a primary structure of amino acid sequence, then it folds upon to form a secondary structure with the alpha helices and beta sheets. Then it folds upon itself to form a functional fold, followed by the multi-component system or the quaternary structure. So this functional fold is formed in the tertiary structure of the protein. Now, similar structures have been found for the RNA also. So, what happens? The primary structure of RNA is basically the sequence, nucleotide sequence. Okay, so that would contain all the like bases connected through phosphodiester bond. that will fold upon itself to form a secondary structure, and that will fold upon itself, forming a functional fold in the tertiary structure. So that this is structure is common, or it's similar to that of the protein molecules. Okay, so now we'll discuss a bit in detail about the structural hierarchy of the RNA. So going to the primary structure, it's a chain of nucleotides formed by the like attached to the phosphodiester bond. So it will have A, C, U, and G connected with each other in a sequence, and the sequence can be like basically derived from the DNA. So DNA encodes any of these sequences, and it will form a sequence that would have a functional role. Now, moving on to the secondary structure. So, basically, as you know, the tRNA ka structure, it is also a clover leaf like structure. So this is the clover leaf structure or the secondary structure of the tRNA. So as the these loops or these stems are formed by the folding and complementary pairing of the nucleotides. So RNA would have some complementary pairing forming the stems. Okay, like we can see here in the example, this is stem 2, this is stem 1, and this is stem 3. So this stem forms the and there would be some complementary base pairing between the substrate as well as the stems. So as you can see, the stem 1 and stem 3 are basically helps in positioning of the RNA to the RNAs or the ribosome so that it can cleave here. And this stem 2 functions in forming this particular site that helps in cleavage. Okay, so this is how the secondary structure of that particular RNA is formed. This is similar to that of what we study in tRNA. Okay, so like this, as I said, this is the secondary structure of the tRNA, and this is the secondary structure of the hypothesized ribosome. And when it folds upon itself, it forms a tertiary structure. Okay, so now the major function of this particular tertiary structure is that it will have a particular site where the substrate will come, and it will have some function, or it is broken down into the product. Sorry. So this gives the tertiary structure of the RNA molecule. So moving on, so why we are telling that the RNA has a catalytic role? That is because, like in enzymes, we see that there would be an active site where the substrate would bind and it will be converted to product, as I just discussed. Okay, so this is the substrate, it will bind and will be converted to the product. Now, this active site is the functional site of the enzyme, and it is governed basically by the sequence of amino acids. So here, for example, if I take the example of chymotrypsin, so it is a protease that cleaves the bond. So here, three molecules or three amino acids like the serine 195, histidine 57, and aspartate 102, these forms a catalytic triad that basically catalyzes the cleavage of the peptide bond. Okay, so this forms the active site of the chymotrypsin molecule, forming the where it helps the cleavage of the peptide bond. And this glycine 193 basically helps in positioning of the protein to the active site. Okay, so this particular active site is formed with the help of these particular amino acids. So the sequence governs the formation of the active site. Similar is the case with the RNA molecule also that we have seen here. Okay, so this sequence governs, for example, if the complementary base would not be there, then this sequence, this stems will not be formed. Okay, and this will lead to formation of an active site kind of structure that is functional in nature and that catalyzes the the cleavage of the RNA molecule from the parent one. Okay, is it clear?
Yes, sir.
Okay, so this is all for this class, and in the next class we'll discuss some of the examples in detail, like RNAs, P, ribosome structure, and all those similar examples, and how these things are like important in real life.
Okay, sir.
Okay, so I'll upload this recording also today and share the link with you, and if you have any doubts you can ask.
Sir, can you please share the recordings of previous lectures
Yeah,
as
I'll
well?
share.
Okay, sir.
Hmm.
Thank you.
Okay.